Hello everyone, I'm Valeria Turina and today I'm going to talk about the topic of botulinum toxin, also known as Botox, and the impact that it has on animal testing. This presentation is structured as an introduction to the botulinum toxin, followed by where it is used, medical and aesthetical industry, also how it can be made, and in the last part I will talk about how it is tested nowadays and how new techniques can be used to substitute the animal testing, and the conclusion in the end. Botulinum toxin is considered to be one of the most poisonous molecules known. In fact, it is estimated that the lethal dose is around 0.1 and 1 nanogram per kilogram in intravenous and 1 microgram per kilogram by oral administration. It has the ability to paralyze both motion and automatic nerves. Due to its severe toxicity, it is very important that each batch is tested to have the same consistency and potency of previous batches. Even if the batches come from the same bulk, it is important to understand the quality batches. Botulinum toxin is a 150 kilodalton polypeptide composed by two specific chains, one light chain of 50 kilodalton and one heavy chain of 100 kilodaltons. Both chains are linked together with disulfure bridge and with a loop as shown in this picture. Botulinum is used in both medical and aesthetical industry with local injections. This molecule can relieve from different types of diseases such as muscular spasticity, post-stroke spasticity, head and neck spasms, strabism, hyperactive urinary bladder, excessive sweating, migraines, and clenches of muscles such as esophagus and jaw. It is also used with aesthetic purposes, including the reduction of facial wrinkles. Botulinum toxin is produced by Clostridium botulinum, which is a gram-positive bacteria that are capable to produce the toxin. It is known that at least 7 different types of botulinum toxin can be produced, but the most common used are type A and type B, and they are identified as poisoning for the human body. The toxicity of botulinum is due to the interaction with SNAR proteins that mediate with neurotransmitters released of synaptic vesicles in neurons. If botulinum binds to the receptor, the acetylcholine cannot anymore, and the response of muscle cells is stopped. To test the efficacy of botulinum toxin, animal testing are the most widely spread, which is resulting in the sacrifice of hundreds of animals every single year. Approximately 400,000 animals are used in the EU to test botulinum toxin. The hole in the bureaucracy is that animal testing for beauty purposes is not allowed in the EU. But companies claim that they don't know if the product is bought for medical or aesthetic purposes. Mouse lethality bioassay (MLB) are the most common tests used to evaluate the potency of botulinum toxin. LD50 assay is the only method used to test safety and potency of every single batch of botulinum toxin. In this assay, different doses of the toxin are injected into the mice. It is reported that between 5 and 10 picograms of milliliter is the lethal dose for a mice. The main effect of botulinum on mice is asphyxiations in the first 24 hours of the most affected mice, and also the paralysis of the lower body, that sometimes can also kill the mice, of dehydration and weight loss because they cannot reach food and water. The 3R concept is in disagreement with this practice because the animals are under great distress. The test can last from 24 up to 96 hours, and only around 20% of animals die in a human way because of the reluctance to kill the mice too soon due to not have accurate results. However, the mouse lethality bioassay is not effective in the detection of botulinum spores, so the accuracy and the possibility to have false positive results is high. The error rate is estimated to be around 40% due to the high variability between laboratories. The main problem with animal testing are the severe suffering of the animals and the high number of animals used. Already, some companies do their testing in vitro, but it is almost needed to test also in vivo. Other types of testing are now being implemented. For example, ex vivo assays, cell-based assay, and also human-induced pluripotent stem cells assay. Ex vivo assay consists in a continuous measurement of the twitch force stimulated by electricity in an isolated explant of an adult rodent muscle and nerves. With this method, Botulinum demonstrated a dose-dependent decrease in the concentration amplitude of the stimulated muscle. This assay can replace the MLB, but they still use animals. Cell-based assay can be used to test botulinum, and they are created with specific cell lines that include membrane receptor binding and toxin uptake. One of the most used is the SNAP25 that can be qualified with a Western blotting, ELISA, or immunofluorescence assay. These cells can be used without limitation and can be produced in large quantities. The main problem is that the sensitivity to the botulinum toxin can vary from different cell lines, and sometimes they fail to duplicate many of the neurotypical properties seen in mature neurons. The most promising test that could be used for botulinum toxin is the human-induced pluripotent stem cell assay. The use of these cells of human origin should increase species-specific release and offer higher sensitivity together with the possibility to compare different batches of botulinum. The neurons from these cells express all the necessary receptor and substrate for the monitoring of the toxicology. This method is not being used yet because more optimization is needed. In conclusion, it is possible to see that majority of the testing is still being done with the use of animals, but in future years, due to the EU laws and the awareness on this topic, other techniques will be starting to be used more often for testing of botulinum toxin. The main problems with the new techniques is that they require specialized personnel to implement the assay, and the reproducibility and stability of the assays are not so clear. But the same it can be also said by the use of animals in different laboratories. The replacement of animal testing has been progressing since 2011, but the difficulties with validating the alternative methods creates a harder transitions. I thank you everybody for the attention. Any questions?